This talk is about swimming in the third domain. It's like, I guess, what it says on the title slide. And this title slide, I like to open on a nice, pretty picture. This is the Great Salt Lake in Utah. In fact, this is Antelope Island, which is an island set in the middle of the lake. The silhouetted figure in the foreground is my poor, long-suffering wife. This is when we were doing some field work over there, looking for extremophiles living in the Great Salt Lake. And this project is a collaboration between people from the University of York and from Westminster College, which is in Salt Lake City in Utah. So people who don't know me, Dave gave quite a nice introduction. My lab is based around biological and soft matter physics, and particularly we're interested in novel imaging techniques. The philosophy, I guess, is to build small bits of equipment that are quite simple, but you can take them into nasty places to do interesting experiments. So the picture on the screen on the left is my lab here in York. Well, that was my old lab. I've recently moved labs across campus, the same department, we've got a new building. This is a holographic microscope setup, and I'm going to talk a little bit about that in a minute, but I'm not going to focus on techniques today. And some of the applications we've been investigating recently are leishmania parasites, which is a nasty sort of flesh-eating tropical parasite. There's a BioArchive paper up at the moment. We're currently touting it around various journals, trying to get someone to bite. We're also interested in halophilic archaea, which I'll introduce more formally in a minute. We're interested in predatory bacteria, and we're growing some predatory bacteria, and I've got a couple of PhD students, and I think I saw Eric Rodriguez amongst the audience, and he's working on that. And we're working a little bit on chlamydomonas algae as well, but that's had to be put to one side again for a little bit because of the lab moves and everything else, but that's definitely a going concern. So the pictures at the bottom of the screen here are pictures of some of the sampling locations that we've been investigating. And this is Bulby Potash Mine, which is one of the deepest working mines in Europe. I think it might be the deepest working mine in Europe now. And this is one of the places that we're going to have a look for weird organisms that we can study and examine the swimming of. So the technique we use, as I mentioned, is holographic microscopy. And this is high-speed 3D imaging. What does that mean? I've given a couple of examples of applications that we've used the technique for so far. Typical volumes that we would study, 1.5 millimeters on a side, measure a cube. We can sample 500 volumes a second, and then measuring very fast phenomena, we've measured sort of 60 microns, sorry, that's 60 millimeters, that should say 60 microns on the side, and we can image at 120,000 volumes a second. So that's for some physical science stuff rather than biological stuff. So we're interested mainly in swimming cells and what we can find out about the biology of swimming cells based on how they're swimming. Look a little bit at the flagella of cells as well. Some of the applications I've listed over there, we've published on malaria microgametes back in 2013, more recently on this species, Shewanella putrefaciens, which some of the people interested in swimming might have seen in the literature recently. It's a bacterium that swims, and then if it backs up, it can wrap its flagellum around its body and form a little corkscrew to get out of tight spaces. That was interesting. Done some work on active colloids as well, sea urchin sperm cells, E. coli, and most recently on halophilic archaea. So that work on archaea has been accepted, but it's not coming out until the middle of September, I think, as Dave mentioned. So apologies for anyone for whom this is really dull, but I wanted to introduce the bacterial swimming in chemotaxis because I'm going to contrast archaea to this in a minute. So bacteria swim around. We know this because we can look under a microscope. And we know that bacteria swim thanks to people's experiments with cryo-EM microscopy, electron microscopy and such like back in the 70s. People can see that bacteria have a helical flagellum out the back of the cell. And we've got a cartoon of this at the top right-hand corner of this slide. This helical rigid flagellum is driven by a rotary motor, and this pushes the cell forwards. And people discover this, as I say, only really through the use of electron microscopy. The flagella are too small to be seen by optical microscopy unless you play really funny tricks. On a good day, you can see flagella with DIC microscopy, but you've got to work really hard to see them. You can fluorescently stain them, which makes your life a little bit easier. So the erratic motion of individual bacteria has been studied for a long time, but the real sort of step change in the understanding came with this classic paper from Berg and Brown, and I've got a screen capture of that on the bottom right-hand corner of the screen. And Howard Berg built this fantastic tracking microscope, which he's still using sort of 50 years later. The idea was that you'd have a sample stage, and the microscope would be focusing through the sample volume, and by complicated arrangement of optical fibers and photodiodes and that sort of thing, you could teach the microscope to follow cells around. It could move the stage by keeping the cell in the center of the field of view. By reading off the stage position, you could track individual cells in 3 dimensions. And This paper it's a great paper. It's about 4 pages long, and it set the template for a lot of the work that came after that. And Howard Berg noticed that cells run and tumble in 3D. So run in a straight line, then change direction, run in a straight line, so forth. And at a large scale, this gives rise to a random walk. In fact, if bacteria are doing this random walk in the presence of a chemical that they like, they can extend their run duration as they swim up the chemical gradient, so towards the source of food. And this biases the random walk and allows cells to drift. So the physical considerations, which don't often get much of a look in, of course, Brownian motion dominates life on the micron scale, and it randomizes swimming direction at all times. There's a curious fact I'm going to come back to later. This is Purcell's scallop theorem. Of course, a lot of people here will be familiar with low Reynolds number and know more about low Reynolds number hydrodynamics than I do. But the idea, of course, is that if you have a reciprocal motion, like a clamshell that's opening and closing, this reciprocal motion, this 2-step reciprocal motion, doesn't get you anywhere. There's a cute paper back in about 2011 by Eric Lauger, in which he noticed that if there's a source of noise in the background, Brownian motion can break symmetry. In fact, there's an advantage to doing reciprocal motion in the presence of Brownian motion. There's a graph from Eric's paper in the bottom right-hand corner of the screen. This is a mean square displacement for a non-swimming cell at the bottom, a ballistically swimming cell at the top that eventually has its swimming direction randomized by Brownian motion, and then this reciprocal swimmer, which actually achieves an increase in mobility despite the fact that classical low Reynolds number hydrodynamics says it shouldn't benefit from that kind of reciprocal motion. So Howard Berg constructed some kind of heuristic arguments around the behavior that he saw in his microscope. And he said that because ballistically swimming E. coli swimming in a straight line, has its swimming direction randomized in a few seconds, they must tumble roughly once every second to kind of defeat this motion. So if they're sampling different directions and occasionally extending their run in a favorable direction, they can overcome the randomizing effect of Brownian motion. So that's a picture that's governed most considerations of swimming cells on the micron scale, certainly bacterial cells. By the time you get to bigger things like Chlamydomonas, the rotational time scale is longer, you start to have different rules, but for bacteria, Berg's kind of heuristic arguments have held up very well. So I'm not going to go into a lot of detail about how bacteria navigate, but I wanted to put up a couple of slightly scary looking pictures. Because I'm a physicist, I can get rid of most of the detail here. This is a great review. If you want to know anything about how E. coli and how B. subtilis and these other cells do chemotaxis, check out the review by Stephen Porter, George Wadhams and Judy Armitage. This is a simplified model of a bacterial cell. So there are 3 diagrams on this picture. The top one is the cell body with the flagella motor and the sort of chemosensory apparatus, and at the bottom is a close-up of the base of the flagella motor. But I'll get rid of the motor to start off with, and I'll kind of simplify it down to the important parts for this talk. So E. coli have a nose at one end of the cell made out of receptors, and when attractants bind to the receptors, something happens inside the cell. Now, usually there are these chemical messengers, these messenger molecules called key Y molecules that just float around, and sometimes they get a phosphate group plugged onto them. And there's a kind of equilibrium level of phosphorylation and dephosphorylation. When the cell detects something that it likes, the phosphorylation is suppressed, so fewer of these messenger molecules have a phosphate group on them. And when I was talking about a bacterial nose, incidentally, I wasn't being just whimsical about it. In fact, cryo-electron microscopy, and you can see this in the picture at the bottom of the screen, has revealed that these receptors form crystalline arrays at one pole of the cell. So we can start to think about diffusive timescales within the cell and that sort of thing, because we can actually map the anatomy of an E. coli cell. And in fact, this receptor complex is remarkably well conserved. At the bottom there, you might not be able to make out the writing, but there's E. coli, Salmonella enterica, Bacillus subtilis, a number of different bacteria that all have this crystalline array of receptors at one end of the cell. And these receptors, as I say, are responsible for ultimately plugging phosphate groups onto messenger molecules. In the case of E. coli, when an activated molecule, when the phosphate group is on the key Y can stick to the base of the molecular motor, and this makes the motor more likely to tumble. This is a probabilistic effect, but the more phosphorylated key Ys, the more activated messengers are plugged onto the motor, the more likely a reversal is. So if the cell is swimming up a gradient, the messenger molecules are switched off and the probability of a tumble drops. So there are also, I've uncovered a lot of this sort of adaptation mechanism. There's a whole load of stuff, there's these key W and key A proteins. So essentially the E. coli cell can tune the sensitivity of its chemosensory apparatus. It can make it more sensitive if there aren't many molecules around, or if it's traversing a steep gradient, it can make the apparatus less sensitive so the detection system doesn't saturate. And this is pretty well understood in E. coli. One of the reasons that E. coli is studied so much for chemotaxis is because it's got quite a simple chemotaxis system. There's really just one pathway mediated by this key Y molecule. Other species, sort of familiar model systems like Pseudomonas and Rhodobacter, have far more key Ys, and then they kind of start to compete to bind with different bits of motors, and things get much more difficult. But E. coli just has this one key Y, which is why most of what we know about chemotaxis is kind of based on that viewpoint, I suppose. But life isn't just about bacteria. So this is a paper from 2016 by Hug and co-workers, and this lays out a new proposal for the tree of life. So at the center of this diagram, the center of these radiating lines, there is the last universal common ancestor, or LUCA, some people call it. Now, the top of this tree is all of the bacteria. Bacteria are fantastically diverse, and this is a genomic type construction that describes how different species or different phyla are related to each other. Everything, as I say, at the top there is a bacterium. I wanted to highlight one particular region of this bacterial tree, and these are the gram-positive bacteria. This means that the outer membrane of this small subset of the bacteria is quite distinctive. They don't have an outer membrane in the sense that most bacteria do. The gram-negative bacteria have an outer membrane. The gram-positive only have an inner membrane and a bunch of stuff around the outside. And this is thought to be an early divergence between two different families of bacteria, the gram-positive and gram-negative. The reason I'm making a big deal about this is because archaea have a chemosensory apparatus which is very closely related to the gram-positive apparatus. But I haven't got to archaea yet, so I'll back up a little bit. So the rest of this tree, the bottom right-hand corner, we've got eukaryotes, so protists, plants, animals, fungi, trees, bananas, all that good stuff. Then archaea, the bits that I've highlighted in red at the bottom. So in the 1970s, these are recognized as being different from bacteria because you can start to use the tools of molecular biology to pull cells apart, find out what they're made of. And they're recognized by Carl Woese as a completely different branch of the tree of life. Some people are saying that these are more ancient than both bacteria and archaea. Some people say that archaea are good candidates for what eventually became mitochondria in animal cells, but I'll leave that to biologists to fight over. The important thing is these ancestral organisms are ubiquitous, but this wasn't recognized for a long time. Often people would only recognize them for very extreme environments. So you find a lot of archaea in hydrothermal vents at the bottom of the ocean, or salt mines, or extremely acidic places, extremely hot places, this sort of thing. Now, a lot of bacteria swim fast. And going back to the this idea of swimming and swimming being important for life, I've got a sort of a world record chart of different bacterial swimming speeds there. So E. coli is relatively sluggish, but still manages 10 body lengths a second. Some of the ocean-going bacteria, like this Vibrio species and this Thiovulum major species at the bottom, swim very, very fast, or almost 0.5 millimeter a second, despite being only a few microns to a couple of tens of microns in size. The video is an example of Serratia marcescens swimming around on the surface of an agar plate. This was made by a former postdoc of mine in the lab. So bacteria swim pretty quickly, and so do some archaea as well. So a lot of the methanogens swim quite quickly, and archaea are the only source of methane, I believe. I didn't know this. Occasionally these facts pop out in bacterial and biological conferences. Some of these methanogenic bacteria swim very quickly. And the thing that I found most interesting about this is that I've got an electron micrograph of a type of archaea at the top right-hand corner of the screen. And in fact, the propulsion system that archaea use is almost identical to the bacterial one superficially, but if you look at it in detail, it's evolved from a completely different structure. So the filament is helical, sticking out the back of the archaeal cell, but it's made of a different type of protein to the helical filament sticking out the back of the bacterial cell. Archaea use a rotary motor, but it's powered by a different power source. It uses ATP. Bacteria use a proton gradient. Life has effectively arrived at the same engineering solution to making cells move twice by two separate pathways. So this has got interesting ramifications. You know, if we start looking for life in other places, we start building space probes and everything else, maybe we should start thinking about looking for things that are spinning, because that seems to be a very, very interesting way to get cells move around, and it's occurred twice on Earth to start off with. So, food for thought. Anyway, not all archaea swim that quickly. In fact, a lot of them swim quite slowly. So some of the best understood archaea are these halophiles. So these are salt-loving archaea, and they live in places like the Dead Sea, salt mines, the Great Salt Lake, and these are the ones that I'm going to talk about for the rest of the presentation today. So the picture at the bottom left-hand corner here is a picture published by Kinoshita et al. This is from Nishizaka's lab in Tokyo. And they found this rotary motion. They measured the rotary steps the archaeal flagellar motor undertakes, and they found that they swim very slowly. In fact, all of the studies on halophilic archaea seem to find very slow swimming. So the cells swim at maybe half a micron a second, 1 or 2 microns a second, that sort of speed. So we went off looking for these. The best place to find halophilic archaea, one of the best places, is the north arm of the Great Salt Lake. And this is a nice picture of the north arm of the Great Salt Lake in the middle of the summer. The water is pink, and this is largely due to the sheer quantity of archaea that you're looking at. So archaea express bacteriorhodopsin as a way of protecting them against sunlight. They get a lot of UV radiation in the Great Salt Lake, and the entire lake turns pink. Of course, I went there in January when it was absolutely freezing cold, with my microscope built out of drill batteries and all the rest of it. It was absolutely freezing. The only reason the water is liquid is because it's Salt Lake, so obviously the freezing point is depressed. Snow all around at that time of the year. Next time I'm going to go back in the summer, I think. We managed to collect a few samples, and our colleagues over there, Bonnie Baxter and Jamie Butler, kind enough to send us some samples on plates a few months later, and we've entered into culture some species that we found in the Great Salt Lake. When I came back to the UK, I was also aware of a project that was going on in Bulby Potash Mine, which, as I say, is about an hour or hour and a half north of York. It's the deepest working mine in the UK. And in the back of this image on the right-hand side, you can see what is in fact a particle physics lab. So this is where people are searching for dark matter. And the reason you do this in a mine is to get away from all these horrible ionizing particles from the sun. But it's interesting because it's a salt mine, and occasionally you get salt water seeping through the walls. And I'm interested in what lives there. Whether or not you believe it, some people claim that organisms can be trapped in brine inclusions in salt for a very long time. There is some evidence, again, I'm reluctant to nail my colours to this mast, but some people have suggested that cells can stay dormant in there for millions or hundreds of millions of years and then be revived afterwards, which poses some interesting questions about how robust life might be in the long term. So this is one of the salt crystals that I extracted from Bulby Mine. In fact, I took some salt water from a puddle that I found and I let it crystallize. And what you can see, these lovely white lines, are due to the fact that we're looking through a salt crystal using dark field illumination. There's a sort of square feature at the bottom, which is the edge of a salt crystal, and these geometric patterns are caused by water inclusions, supersaturated water inclusions in salt crystals. And you can also make out some pink features jammed into the edge of the crystals, and these are the cells that we extracted. Very good. We find these pink-looking things from the mine. The question is, do they swim? This is an excellent question. I'm going to play a movie. You probably won't be able to see much, but that's because there isn't much to see in this movie. To give you a sense of scale, this is a dark field movie, and the scale bar you can see there is 100 microns. So we've got a couple of specks. Some of them are out of focus, and they're kind of shimmering a little bit. The question is, are they swimming very slowly, or are they just diffusing? It's kind of difficult to tell, but what you can do is take a video sequence like that, and if you color the different frames, so the frame 1 is red, and you colored each frame through the rainbow through to violet at the very end, you do some background subtraction, all the rest of it, you can obtain a picture that looks like this. So this is a colored time-lapse image, and the rainbow colored streaks show you where there is a cell swimming through the environment. Those little white splodges are Brownian diffusing either non-motile cells or specks of dirt that we've picked out of Bulby Mine. We've got cells swimming in there. There is something living in the stuff that we pulled out of Bulby Mine, and in fact the same is true of the stuff from the Great Salt Lake as well. It's two very diverse locations. What are they? That's an interesting question, but this is something that's become a lot easier in recent years. So whole genome sequencing is now available, and my PhD student, had a very good PhD student on this project called Katie Thornton, who's now graduated and moved off to different things. And she was able to take these isolates from the Great Salt Lake and from Bulby Potash Mine, streak them out on agar plates, take single colonies, grow up the single colonies, and then send them after a bit of processing for gene sequencing. I think it cost about £70 per strain, and they give you the whole genome. Because Katie had done a really good job of the purification, we were able to read out really nicely what we had living in our samples. And the nice thing is that these are both archaeal samples. And I've shown part of the sort of the gene assembly at the bottom of this picture. Halopharax species from Bulby Mine and Haloarchaea species from the Great Salt Lake. And because we can read off the whole genome, we can find what the different components of the cells are. And we can see that not only do they have motors, as we would expect from motile archaea, we can also see where they've got chemosensory stuff floating around. And this is one of the key points, and the reason that I was going on about bacterial chemotaxis earlier on, it's because a lot of archaea, particularly salt-loving halophile archaea, have these key Y proteins, exactly the same as you see in gram-positive bacteria like Bacillus subtilis. The working hypothesis is that these were acquired by horizontal gene transfer. But if you remember, this is fascinating stuff because bacteria have a completely different flagella motor to archaea. So it's like opening the bonnet of a diesel car and finding components from a petrol engine in there. Maybe you'd expect it, maybe you wouldn't. It's still quite a striking feature, especially because the motor in an archaea is different to the motor in a bacteria, and it's surprising that the parts fit. In fact, there are adapter proteins at the base of an archaeal motor that we think allow them to interface with bacterial type key Y proteins. But our archaea both have key Y, and that motivates the use of a particular chemotaxis model later on. This is what the cells look like under DIC illumination. There's not really much to see. They're small, vaguely E. coli looking things, but they're about half the size or half the volume, something like that. So I use a couple of abbreviations later on in the talk. I've got HXBM from these cells that we've fished out of Bulby Mine, Halopharax Bulby Mine, and the Great Salt Lake, we've got HGSL, that's Haloarchaea Great Salt Lake. So we use the holographic microscope to track these cells in 3D and see what we can say about their chemotactic performance. So we image an area about 1 millimeter squared by about ½ millimeter in depth. And we take a few different movies, we make different biological replicates, all of that stuff. So we track about 2,500 cells from each of our strains in total. And we can sort these strains to extract the cells that are swimming from the cells that are just diffusing. And the cells that are swimming, we can see because they've got a characteristic mean square displacement that scales with time squared, as opposed to diffusing cells, which have a mean square displacement that scales with time. So it takes about 30 to 60 minutes to process a video, and we end up with about 200 megs of track data in total for about 100 gigs of video data. So what do we see? Well, we find there's a distribution of swimming speeds. It's kind of vaguely Gaussian, but they all swim very slowly. Something like 2 microns a second is the mean swimming speed. We can pick out the distribution of tumble angles because we've done that before for E. coli, and we know that we can do it properly. It turns out that our archaea simply run forwards and then reverse. And they don't do any of the clever run reverse flick, which is what you see in some ocean going bacteria. They simply move forward slowly and slowly reverse. So it looks like they're taking advantage, they're exploiting Brownian rotation to help them change direction. They don't change direction on their own. They don't reverse terribly often. This is a little bit surprising. So the mean run duration for both of our species is greater than 10 seconds. It's sort of in the region of 13 seconds. And we wondered if that was just us, but actually if you poke around in the literature, not many people have looked in detail at run duration of halophilic archaea before, but there are a couple of references, and Reinhard Wirth and Dieter Oesterheld, some authors in Germany, have put quite a lot of work into this, and particularly the biochemical parts 10 or 20 years ago, and they found data that sort of corroborates what we find. So we think this is a fairly universal feature of halophilic archaea. So this is the puzzle. People at Linda Turner, who's working in Howard Berg's lab, has tracked B. subtilis and found that the cells tumble once every second, more or less on average, and that's a bit like E. coli. B. subtilis reorients due to Brownian motion every 3 seconds or so. And the paradigm in the literature is that for E. coli, a run duration of even 5 seconds would be prohibitive, i.e. Brownian motion would randomize its swimming direction more quickly than it was reversing, and it would lose information on where it is. That would all be washed out by the effect of rotational Brownian motion. Now we have much smaller cells that reverse every 15 seconds. They also reorient due to Brownian motion on a time scale of, well, 1.5 seconds if you believe the arguments based on cell size. And they use a key Y molecule, a chemical sensing molecule, which has been inherited from these bacteria. So this is a bit of a puzzle, really. You would think that chemotaxis doesn't work based on these heuristic arguments for our cells. So we're doing what physicists do when they get stuck, and we run some numerical simulations. So what we've already obtained are the swimming speeds. We've already obtained the rotational diffusivity of cells. What we do, sorry, a big one, we've got the run duration of cells, and what we don't have is the rotational diffusivity. So we need to measure how quickly the cells wander off course. We can do this by measuring using the 3D tracking data we've got. We have to fit a slightly more complicated model than simply an exponential direction decorrelation model, which is what you'd expect kind of naively. You can get this from polymer physics analogs, this sort of thing. So our cells move with a slightly helical motion, and we've got to build that into the direction correlation model we use. This will all be in the paper. I'm happy to talk about it later, but I don't really want to dwell on it. If we fit this model to our data, we can reliably extract the rotational diffusivities. So we find that the cells take about 6 seconds to wander off course. That's the characteristic rotational diffusion time, which tells us that the cells are stabilized by the flagellum or flagella at the back of the cell. They're stabilizing the cell against rotation due to Brownian motion. But we've got numbers there, so we can start to create simulations. So we can say, okay, so our archaeal cell, well, it crosses our field of view in a few tens of seconds, but that's not really long enough. These things are living in the Great Salt Lake, deep in undisturbed waters, or they're living in Bulby Potash Mine, where they live in brine pools, and they can be there for very, very long times. We can do simulations for very long times, sort of days at a time. And we're going to take the limiting time scale of our simulation. Although we simulate for a very long time, we say the longest useful time scale is going to be something like the cell doubling time, which is a few hours for our archaea, and that's in fairly optimal conditions. So we simulate loads of different cells. We vary the swimming speed of cells. We vary the run duration. We put the cells in a simple linear chemical gradient, so we're not playing with time-dependent gradients or anything like that. We borrowed a chemotaxis model from E. coli studies, and the reason we feel justified in doing that is that we have the genomic data that tells us the cellular components responsible for chemotaxis in archaea are similar to those in bacteria. And we can simulate these different things. The tumble rate, incidentally, if people are interested in exactly the nuts and bolts of the chemotaxis model, the central equation is down there at the bottom. And this tells us there's a tumble rate, lambda, which is modulated by the cell's recent history. So I've got an integral at the bottom right-hand side. That's a convolution of a response function, and C of T prime, which is a concentration that the cell has experienced in its recent history. The response function has a finite duration, so that means the cell has a memory length of about 2 seconds. And we tried playing with the memory length a little bit to see how that affects the chemotactic behavior. That's in the supplementary materials of the paper. You're welcome to go and have a look at that when it comes out, or I could talk about it afterwards. It turns out it's not that interesting. Something like 2 seconds, 1 second, 2 seconds seems likely, given the size of the bacteria, given the diffusion coefficient of the proteins and that sort of thing. And that's the one we've used in the simulations. So what we find, we express the chemotactic drift speed as a fractional speed. So Vx is the speed the cell travels up the gradient, divided by V0, which is the swimming speed of the cell in a straight line. V0, of course, is what we put into the simulation, and Vx is what we read out. This is a way of comparing cells that swim at different speeds, of course, and I'll get to that in a little bit. So the results here in the bottom left-hand corner are what happens if you vary the baseline run duration. So our archaea, if you remember, have a mean run duration of about 13 seconds. This graph shows what happens if that wasn't 13 seconds, but if it was different lengths of time. So we've got 3 different swimming modes. We've got some cells that both lengthen and shorten their runs. We've got cells that lengthen their runs only. We've got cells that shorten their runs only. So the cells that lengthen and shorten their runs can extend runs in positive directions. They shorten their runs if they don't like what's happening. So if they're noticing a drop in the local chemoattractant concentration. The cells that lengthen their runs only are thought to be like E. coli. So the working model is E. coli has the default run duration if it's heading down a gradient, but it has longer runs if it's heading up a gradient. B. subtilis and these gram-negative bacteria, the gram-positive bacteria, have the lengthen and shorten run type model. The shorten runs only model was put in there for the sake of completeness. And what we find is that a run duration of something like 8 to 10 seconds is more or less optimal. Interestingly, the cells that shorten their runs only become more effective as the run duration, the baseline run duration increases. So this becomes a more effective strategy, sort of changing direction if life is getting worse becomes a more effective strategy if your baseline run duration is longer. So at the bottom right-hand corner of this slide, you can see the mean squared displacement of cells running at different speeds as a function of delay time tau. And I've taken mean squared displacements for each of the cells showing at different speeds, and I've scaled them by 1 over tau, 1 over time. So a horizontal line on this graph denotes purely diffusive behavior. A slope of 1 denotes swimming behavior in the bottom right-hand corner. So the cell that doesn't swim at all, so V equals 0, and in this set of simulations on varying swimming speed, the cell that doesn't swim at all, of course, just constitutes a straight line when you look at its mean squared displacement. I've included a couple of coloured bars that correspond to the diffusivity of different molecules, different molecular weights. So glucose is a tiny little molecule, and that has the highest diffusivity and corresponds to the light blue line across the graph. What this tells us is something that we don't necessarily feel surprised by. So at short times, cells display a ballistic swimming behaviour. At intermediate times, you start to see a diffusive behavior, which corresponds to a random walk. And then at longer times, you start to see the effect of a biased random walk as cells start to drift up the chemical gradient. The reason this graph is here, all cells at all finite swimming speeds will eventually be able to drift up a chemical gradient if we assume that the gradient doesn't change with time. But the time scale for this drift up a gradient is important. There's a fairly sort of aggressive log scale on the bottom there. You'll notice these are 2 orders of magnitude at each tick. And what we find is that if you swim at 2 microns a second, you can just about catch up in some sense with small molecules if you're swimming at 2 microns a second. So with that, we of course thought, great, this is a nice result. We sent it in for peer review, and of course the reviewers very politely pointed out this is a very nice peer review process. I'm not sure if the peer reviewers are in the audience, they may be, but actually it was a very positive experience of peer review, and the paper is a lot better as a result. The reviewers kindly pointed out that we hadn't actually demonstrated that our species perform chemotaxis, which is an excellent point. So we went away and we did some experiments. We find out what we do. To do the chemotaxis experiments, we mixed methionine into some agar and plugged one end of a sample chamber with this methionine infused agar. Why did we choose methionine? Well, some people have done some agar plate assays which have their drawbacks. You can't always state definitively that chemotaxis is going on with agar plate assays, but it's a starting point. And actually we find that our archaea did assemble in a kind of chemotactic band close to the interface, or set back a little bit from the interface of our methionine infused agar in our chemotaxis chamber. So by looking at one of the edges of our chemotaxis band and by measuring the run durations either up or down the chemical gradient, we were able to estimate the chemotactic drift speed using this kind of heuristic model that Ahmed and Stocker put in Biophysical Journal back in 2008. And what we find is that both of our strains exhibit a drift velocity which isn't miles away from what we see in our simulations as a kind of optimal drift velocity. Our Vx over V0, so the speed the cell moves up the gradient divided by the underlying swimming speed, is about 0.1 in the case of Halopharax from Bulby Mine, and it's a little bit slower in Halopharax from the Great Salt Lake. To construct these chemotactic drift speeds, as you can see in the panels on the left-hand side, there's this kind of pie chart. The idea of this graph is that the methionine concentration increases from left to right, so schematically speaking, and what you can see is the different segments of the pies correspond to cells swimming in that direction. So cells swimming up the direction have longer run durations, as you can see by the numbers within each pie segment. Generally speaking, slightly shorter runs down the gradient, but that's not as pronounced an effect. And cells running perpendicular to the gradients have what we call the default run duration. This is slightly longer than we see in our previous case because we're running these experiments in a depleted medium. So our previous tracking experiments were done in quite a rich medium. This is done in quite a poor medium, but with the amino acid overlaid over the top. So using these experiments, we can measure what the chemotactic drift velocity is, and we can relate that to other effects. So we've already found that the baseline run duration that our cells exhibit is about optimal in terms of drifting up a chemotactic gradient. We've seen that swimming slowly is quite a good idea, but I was interested in investigating that in a little bit more detail. So the graph at the bottom left-hand corner of the slide here is the result of what happens, and this is a bit of a surprise to me, what happens if you increase the cell swimming speed, only the swimming speed, and you look at the fractional drift velocity up the chemical gradient. And what we find is at very low speeds, there's something about the interplay between cell reversals and translational rotational Brownian motion. Rotational Brownian motion is probably the key there. That says that at very low swimming speeds, the fractional drift velocity is quite low. It then increases up to about swimming speeds of 1 or 2 microns per second, whereupon it saturates. And the value of the fractional drift velocity is about 0.1, so about a tenth the speed of the cell's raw, a tenth of the raw swimming speed of the cell, and that corresponds to what we see in experiments for this strain that we get from bulby mine. The strain from the Great Salt Lake has a slightly lower fractional swimming speed, but it could just be that methionine is not a great attractant to the Great Salt Lake species, we don't know. But what we see from the bulby mine species is roughly the optimum that you'd expect to see if the underlying model of chemotaxis using the keywise and everything else is found. There's also a knock-on effect for this on swimming efficiency. So swimming efficiency, and again, there are heuristic arguments from Berg and other people to say that efficiency will scale as 1 over v squared if v is a swimming speed. So if I divide through the results that you see on the left-hand panel by v, you'll see a 1 over v trend at high swimming speeds that kind of plateaus out at low swimming speeds. So the swimming efficiency drops significantly at higher swimming speeds. This means that it's more efficient to swim slowly, which is a good strategy if you live in a nutrient-poor environment. So bulby mine is certainly a nutrient-poor environment. The Great Salt Lake is not always a nutrient-poor environment, but goes through long periods in winter when it is. The sort of ephemeral patches of nutrients that might be quite long-lived because the brine shrimp appear in the Great Salt Lake. They all sink to the bottom and eventually the nutrients are consumed. But swimming efficiently seems like a good strategy in a hostile environment, and our results from simulations seem to recapitulate this. So this is about the end of what I wanted to talk about. And these are the take-home messages. We say haloarchaeal swimming phenotype is quite consistent. So it's not just us that see these very slow swimming cells that reverse rarely. The same is true for a lot of different species studied by different groups. But no one's really commented on the fact this doesn't seem to make sense in the context of what we know about bacteria. In fact, they perform chemotaxis quite efficiently according to the simulations that we've conducted. And again, we've backed up these simulations using experimental data. We find that the cells swim fast enough given their doubling time to catch things that are diffusing steadily. And Brownian motion seems to have a role in enhancing the random walk. The cells don't change direction very often, but Brownian motion tilts them off course, which also contributes to this investigation and navigation. And in some senses, this is quite similar to Eric Lauber's idea that if you have a reciprocal cell, a reciprocal swimmer that moves forwards and backwards, you can break that symmetry by having Brownian motion on top. Lastly, we say that the simulations based on E. coli biochemistry models seem to agree quite nicely with experiments, and they agree with our genomic data as well. So we're interested in expanding this in future work. And this is what we're doing in the future. So for the next stage, we're looking at building custom instruments to retrieve samples from hostile environments. I'm interested in seeing how the diversity of microbes that live in places like bulby mine, how they all live together and how swimming strategies might differ. And maybe this strategy is optimal. And what you're seeing there is in the bottom window, it might be rather a slow video, but this is me flying a drone in bulby mine. We've got waterproof drones for sample collection as well. There's a picture of the waterproof drone at the bottom left-hand corner. Earlier this year, we achieved our first water landing and water takeoff, so we can deploy and retrieve stuff from hostile bits of the mine where we wouldn't want to send postgrads. We wouldn't even send postgrads in there. You know, that's how horrible they are. So we've got drones to collect this stuff. I'm interested in sampling this further into the future. The bulby mine visits have been very well facilitated by Charles Cockell, Sean Paling, Paul Scoville, the people working out in the mine. They're immensely helpful, and we couldn't have done this work without them. So with that, I'd like to say thanks for your attention. I'd like to thank my postgraduate, Katie Thornton, who did a lot of the genomics work, and this project wouldn't have happened without her. Jamie and Bonnie, who work at the Great Salt Lake Institute, they've been very helpful in giving us the context of the work, as well as the samples and helping our fieldwork trips. Seth Davies, who helped with the genomics, and technical staff, John and Tim, Emma and Eric, who work in the lab now, and thanks for your attention. I'm happy to take any questions.